How is AI going to change the way clinical trials are organized?
So first, let me kind of bring a lot more stuff into AI, because what really is going to change and is changing the way clinical trials are conducted is not just AI, but overall impact of the data analytics-driven innovation, which AI could be part of it, could not. It could be based on classical algorithms, ML, etc. But overall, for me, it's the same bucket. So, and there are a number of ways that data analytics and AI as part of it can affect it. So one of it is basically when we design a new clinical trial today, we are not really using all the accumulated information in the past. So a lot of times we're starting from scratch, and then we're putting a lot of patients, a lot of people to generate the data. So one thing that is very critical before we design a new experiment, a new clinical trial, that we really do our best understanding all the prior data. And then all this prior data should inform the experimental design. So that's one aspect. The second thing is that since there is a lot of data generated now in different places, and that this data is not really connected. So the second thing is to really bring some data into the clinical trial. So for example, what we're doing now, we're trying to reduce exposure to placebo or standard treatment by analyzing prior trials and properly modeling them and bringing this information into the new trial. Which means that if we had to use, let's say, 200 subjects on the placebo, we're reducing this number drastically. So it could be 100, etc., depending on the trial, but it could be drastic reduction of the exposure to the placebo or control treatment. The third way, what we're doing now, we have a cloud intelligence system that has data from many trials, and we're using predictive analytics there. The predictive analytics sometimes is based on machine learning, sometimes it's based on classical methods, but the predictive analytics is trying to predict the response and help us design trials more in a personalized way. A lot of times, what we're trying to do, we're trying to predict the response on the standard treatment, and then basically compare the actual drug versus predicted. So another aspect that can really help is really monitoring the conduct of the trial. So the trials are conducted many times using multiple sites all over the world. So the quality of the clinical sites that are producing this data is largely variable. And very often what happens, the trial accumulates lots of data errors coming from the inexperience or lack of training, or sometimes even deliberate misconduct from the clinical side. So it's very important to monitor the trials so that those errors or those mistakes or the misconduct can be detected in a timely manner to allow for the intervention and changing it. So this is what we call risk-based monitoring, or continuous monitoring of the trials that allows basically to increase the data quality, having a target to ensure the full data integrity of the study, and allow for cost reduction in terms of how we're controlling the sites, how we're doing the audit, etc. So these are some of the things. So there is lots of other stuff that my colleagues mentioned today, more about use of AI in discovery or diagnostics. So, but I think your question was more about the clinical conduct of the conduct of the clinical trials.
So can we say that if we use all these technologies in clinical trials, we'll get more reliable results and trials will be cheaper, right?
So I think the most important thing is the data. If we want to answer some question, we need data to answer that question. And the data is as good as the experimental design can allow it to be. Even if you do more sophisticated statistics, AI, etc., the data needs to be generated in the right way. So if you're doing some of the stuff that I said was really related to the way you generate the data, which should make the trial more optimal and also more ethical, like reduction of the exposure to inferior treatment, etc. Right? The second thing is the data quality. Right? So those two things combined can really make a big difference. I give you examples. So recently, one of our clients is a medical device company, and they had a diagnostic device that got a regulatory clearance this year. So we designed and we did the data monitoring and regulatory submission for that study. So the same company conducted the same trial before in the same patient population and was rejected by the FDA. What was really different in prior and the studies that we participated in and was the study had an adaptive design. It was totally different design, and the study used very heavy monitoring systems at all levels of the device parameters. The device is a complicated device, produces parameters. Under parameters, there is a level of the metadata, so we were doing lots of monitoring for this so that we could separate the noise and errors from the data. So these two things, and monitoring more than the other, brought the same device to the success, which did not happen before.
Today, in many trials, only men take place, and it results in different problems. For example, the dosages that were made for men do not work for women. Do you agree that there is a problem like this?
I think the problem is larger. It's not only men versus women. I think the problem is at the levels that every subject's biological state, so to say, right, is associated with its right dose. So, in other words, there should be right dose for the right patient. So, men, female can be part of it, but the problem is broader. So, I think understanding the personalized treatment regimen is a big problem, and it's a problem that is very is being really addressed and sought a lot right now. So some of the things can be done at the level of the clinical pharmacology, etc., understanding, giving like some testing period and then trying to, understanding the pharmacology at the subject level, not at the trial level, and then trying to go on with this. So the problem can be more complicated. If you have a tablet, for example, it's harder to design the dose. If it's a frequency of injection, it's more doable because you can adjust the frequency of injection. If it's liquid injection, you can, adjust the dose level, concentration, etc. But yeah, I think this is a very important problem because a lot of times the trial can fail because some subset of the study population did not receive the right dose.